Scientists use statistics to allow us to interpret our real data in an unbiased manner. All right, so sometimes interpreting data is really easy. Here's an experiment where a scientist was interested in whether this new food, fish to whale, helps their fish grow larger. So here it's pretty obvious that the new food doesn't really make a difference, and here it's pretty obvious that the new food does make a difference. In these cases, statistics would just confirm what is pretty easy to tell from just looking at the data. However, sometimes you really do need the statistics in order to properly interpret your data. In this case, is there a meaningful difference? It's hard to tell. We would need to use statistics in order for us to really properly interpret this data. All right, so when you're using statistics, you always have an alternative hypothesis or your experimental hypothesis, right, which is what you're really interested in. So fish-fed experimental food will be larger than fish-fed the normal control food, for example. But you need to have a null hypothesis as well, all right, and that denotes equality or randomness of some sort. So in this example, that would be that fish-fed control versus experimental food will not be different in their weights. Okay, and statistical tests always need to have these null hypotheses. So what statistical tests give you as output are p-values. All right, so p-value is actually the probability that we observe data as or more extreme than that observed, assuming the null hypothesis is true. Okay, in another way to state it, if we repeated this experiment over and over and over and the null hypothesis is true, we would expect similar or more extreme results p-value % of the time. So note that both of these are really assuming the null hypothesis is true, what is the chance that I would get the data like what I observe? All right, they are not directly measuring the chance that the null hypothesis is being true. Statistically, you can't do that. All right, don't make that confusion. P-value is not the chance of the null hypothesis being true. Rather, it's assuming the null hypothesis is true, what is the chance that I would get this data? All right, so p-value will be a number from 0 to 1. It's a probability. Large p-values, such as this 0.3, would indicate that your data fit fairly well with the null hypothesis. Therefore, for large p-value, we would not reject the null hypothesis. Okay, we don't accept it because in science you don't prove hypotheses true, but our data would fail to reject the null hypothesis. Now, for a very, very small p-value, right, that means that there's a very small chance that we would get data like ours if the null hypothesis is true. Therefore, we think the null hypothesis is not true. Okay, our data just don't fit with that, so we would reject the null hypothesis. All right, so you fail to reject for large p-values and you reject for small p-values. And what's the cutoff for small here? Well, that varies from field to field, but biologists generally use cutoffs of 0.05 or 0.01. So a p-value of less than 0.01 would be considered statistically significant, and you would reject the null hypothesis. P-values larger than 0.05 would be not statistically significant, and you would fail to reject the null hypothesis. All right, so the statistical test that most of you will be using for your data in these labs is a 2-sample t-test. All right, and a 2-sample t-test, what you're really doing is taking 2 samples, let's say wild-type worms and mutant worms. You do a test, you see how many of them go to the ethanol side of a plate, for example. Or, you know what, let's take our fish example. So here we have our fish-fed control food, here we have our fish-fed the new food, fish to whale. This denotes the average weight of our fish, and here's our plus minus standard deviation. Okay, so control, average, experimental group, average. Well, are these statistically different or not? So we would use a 2-sample t-test, and we would enter in our average, standard deviation, and sample size for the control group and the experimental group. And the 2-sample t-test would then give us a p-value, right? And that would help us determine whether these two groups are different. A small p-value would suggest that these two groups are in fact different. A large p-value would suggest that there might not be a difference between these two groups. All right, now there is also a 1-sample t-test that you might have to use. So for one sample t-tests, you're really using, you're getting real data for one group, right? So here's our average and plus minus standard deviation, but you're comparing that to a theoretical value. So let's say that we were interested in whether worms prefer chocolate or peanut butter. We put chocolate on one side of a plate, peanut butter on another side of the plate, and we look to see which side of the plate those worms go to. Well, our null hypothesis in that case would be that they have no preference. We would expect 50% on the peanut butter side, 50% on the chocolate side. So this value would be right at 50%. And that would be what we would enter into the one sample t-test. And then we would enter our real data, and the t-test would compare whether our real data fit in well with the theoretical distribution, assuming a 50% probability that they'd be on the peanut butter side. It's also important to note that t-tests incorporate standard deviations, right? So here's two examples. Here there's fairly large standard deviations, so the difference between this average and this average are not as meaningful as the case over here where you have much smaller standard deviations, right? It's much more obvious over here that these are statistically significant differences, right? Whereas here, it's not as obvious that they would be. So standard deviation, noise in your data is important and will be factored into t-tests. Also factored in is the sample size. Let's say you just have 3 replicates, right? You have 3 different tanks of fish that you're measuring, and you get this data. Well, what if you get the exact same average and standard deviation, but you did 1,000 tanks of fish or 1,000 fish instead? It's important to factor in how many samples, how many